Chapter 9.4 is going to talk about how the cell cycle is regulated. So again, another big feature, another big theme about AP biology is not that these things just happen in science or in biology more specifically. It's these things are regulated. How do we regulate the cell cycle? So one of the things that happens is we have these growth factors. Obviously, these are proteins that are going to move through like the circulatory system, and they're going to bond with the plasma membrane, and they're going to help make the cell go into the cell cycle or into mitosis more specifically. These growth factors, again, attach to those receptor proteins on the membrane, and they're going to activate some signal transduction to make the cell go through mitosis. This could be because the cell needs to repair itself, or the organism just needs to grow in size. But these growth factors help increase the amount of mitosis going on. Now, again, we don't want all of our cells going through mitosis. We want cells to be regulated. So only about 5% of our cells are going to go through mitosis, while about 95% are going to be stuck in interphase. All right, with these growth factors, you can see they're going to attach to these plasma membranes, more specifically the receptor proteins in these plasma membranes. They're going to create some cascade of signal transduction, which is going to initiate the cell cycle to begin and for the cell to go through mitosis. You can see this is going to be connected to the cell communication chapter. All right, so now we're going to talk about cyclins and CDK, or cyclin dependent kinases. Cyclins are proteins that are going to regulate how much cell division there is in a cell. They increase and decrease during different times of the cell cycle. Really important aspect to know here is that you don't have to know the different CDKs and cyclins that are going to occur at each of the different steps of the cell cycle. That's interphase and M phase. You just have to know what they do. CDKs are cyclin-dependent kinases. So let's break this down. Cyclin-dependent. They are dependent. They require certain cyclins. So these kinases are going to be always present. They're enzymes. They're always present in the cell. They're only activated when they attach to a cyclin. So you can see down here, we have the CDK. It becomes activated when it bonds with a cyclin. That's going to activate another protein. Then it's going that protein is going to activate some portion of the cell cycle. Now, after that activation occurs, this cyclin is going to break down and not work with the CDK anymore, which turns this off or deactivates it. So these CDKs are dependent on the different cyclins within the cell. These cyclins are going to increase and decrease just due to any environmental factors, such as those growth factors we just talked about within the cell cycle. You can see here this chart. Again, don't memorize this chart, but I'm just showing you what the cyclin amount looks like. You can see here within G1 phase, we have the cyclin E. Within the S and G2, cyclin A. Within mitosis, we have cyclin B. And cyclin D triggers just the kind of the whole process here, going from G1 through the S phase here. So these cyclins, again, are going to bond with, let me see if I can go back here. Yes, these CDK molecules, and they're going to activate them, which again activates other molecules, which then pushes the cell through the cell cycle. So you can see this CDK molecule. Again, this is that cyclin-dependent kinase. No cyclin, it's not activated, which means these target proteins aren't also going to be activated. So the S factors are going to be turned off. The CDK here, you can see where we have the G1 and S cyclin. They're going to bond. They're going to interact. Now this CDK is activated, so it can activate other proteins. These proteins are going to make the cell go into the S phase and begin replication. So you can say these cyclins are going to bond with the CDKs. The CDKs are always present. The cyclins are what come and go. Another way that the cell is regulated, or the cell cycle is regulated, is by these checkpoints. These checkpoints are points in the cell cycle that the cell is going to check to make sure everything is working efficiently. And then if it's not, it's either going to fix it, or it's going to send the cell to go through apoptosis. Apoptosis we're going to get to later on, but it's just when the cell dies as a result of some damage or something not going right in the cell. So these checkpoints, imagine like the checkpoints maybe you've heard of in other countries, maybe even here. If you have like a DUI checkpoint, the cops are going to stop the vehicles at night, maybe like on a Saturday night when most people are drinking, and they're going to stop these cars and say, hey, we're just checking to make sure everyone's not drinking. And if you're not drinking, you can pass on. If you have been drinking, they're going to pull you over, and they're going to do like a breathalyzer and check to make sure that you're under the legal limit and that you're not driving drunk. So these checkpoints stop bad drivers that are maybe intoxicated, but they allow the good drivers to move on. So the same way that those checkpoints are going to stop some things but allow others through, these checkpoints are going to allow certain cells through, but not all. Most cells will go through the G1 checkpoint. Most cells will actually go through all of these checkpoints. However, not all cells will go through these checkpoints. The G1 checkpoint, it just makes sure that there's enough room and the organelles have been doubled and there is enough stuff going on in the cell for the rest of the cell cycle to occur. If there is not enough room, there's not enough organelles, there's not enough cytoplasm, the cell will try to fix this first, and then if it can't fix it, again, it was going to make that cell stop in that one cycle and either kill that cell or try to again fix the problem. In our G2 checkpoint, the cell is going to check to make sure all the DNA was replicated and make sure that we don't have any DNA that's damaged. Now, you might think, Mr. Friedhoff, G2, that's not where the DNA replication is going on. Yeah, it's not about the G2, it's about the S phase here. So after S phase, yeah, the DNA will be duplicated, but that checkpoint doesn't occur until G2. So again, S phase is where the replication occurs on, but G2 is where the actual checkpoint is. And again, it's going to just make sure everything's working right with the DNA, make sure it's all duplicated. If it's not all duplicated, it does have fixing proteins that will try to fix the DNA and make sure everything's duplicated. But again, if it doesn't have the right DNA or there's DNA that's just unfixable, it will kill that cell at this point. You might be thinking, Mr. Friedhoff, why would an organism want to kill its own cells? It just doesn't sound right. If it doesn't, this cell then can become cancerous. So a cell that has DNA damage can become cancerous. And the thought there is, I'd rather kill little cells or kill a couple cells every so often than have a cancer form or have a tumor form. Tumors are much more deadly than just killing off a couple cells every so often. You make 300 billion cells a day. Killing a couple here and there isn't going to be a bad thing comparing the overall risk to cancer. All right, the M phase checkpoint is where the cell's going to check to make sure all the chromosomes are attached correctly. They're along the metaphase plate. Everything is just hunky dory, and the chromosomes are in the right places. The cell's in the right places. If not, again, it's going to try to fix it. If it after it fixes it, the cell will continue on. But if it doesn't fix it or can't fix it, the cell will again stop the whole process and try to kill that one cell. But this is just a third checkpoint that you need to know about. So our last part here is talking about that apoptosis like we talked about before. This is programmed cell death. You can see here what's going to happen is the cell is going to fragment and it's going to break apart. This is, again, this is programmed. This is what the organism wants. You can see here there are going to be certain proteins called Caspases that start breaking down internal structures within the cell. Those smaller cellular components here are going to break down then on their own with either more enzymes or with the help of other immune cells from the organism. And again, this is programmed because we don't want this one cell to become cancerous. One cell becoming cancerous can cause a tumor. It can be very deadly for an organism. So it's better to kill a couple cells here than to allow them to become cancerous. You can see here the cell loses contact with neighboring cells, nucleus fragments, membrane develops blisters and gets broken. This is what it actually looks like under a microscope. So this is actually cell death. This is apoptosis occurring in this cell. This is programmed cell death, and this avoids the organism from getting cancer. Very interesting.